Yeah, okay, so we'll start the class right now. I'll just be sharing the screen, so please let me know whether the screen is visible and clearly audible or not. So is it fine? Okay, yeah, it's a thumbs up. Okay, thank you. Yeah, so again, I welcome all of you to the live tutorial session for week 10's content for the NPTEL course Bioinformatics Algorithms and Applications, instructed by Professor Michael Broniha from IIT Madras. So in this particular session, Professor Mumiha, he went over several concepts regarding, for example, docking, 3D QSR, and the related concepts. So we will just talk about some of those, and then we'll try to cover some questions, some numeric questions also, which are pretty similar to some of the questions that we got in the earlier iteration of the course. So hopefully that will be fine. So just as a brief recap, uh, in the last couple of classes, we learned about protein ligand complexes, their binding sites, binding modes, binding free energy, etc. How basically we can characterize the binding in terms of geometry and also the binding affinity between the proteins and ligands. So we talked about that, and then we went over some examples of the binding free energy components and how you can get the total binding free energy and then decompose into different different components which can give you an idea about which interactions are dominating the protein ligand binding and so on. Then again, we talked about the protein folding, the protein unfolded state to folded state, the calculation of free energy of unfolded state to folded state, binding propensities, computative drug design, and ultimately also we talked about some of the physicochemical parameters or guidelines that are very useful while trying to identify whether a small molecule would be a good candidate for a drug or not, right? For example, we went over Lipinski's rule of five. We went over some of those questions, and we also saw basically some of the web servers that can be used to basically predict the aromatic properties for certain small molecules, which can give us an idea about their uh, basically behavior in the physiological systems, right? Then we also talked about the kinetics of protein folding in terms of chevron plot. We looked at different examples and so on. And then we also try to correlate the folding with basically protein folding with different parameters such as contact order, long range order, and so on, right? So basically explaining the folding in terms of the contact order, long range order, etc., which can give us some pretty useful insights in certain cases, right? So these were some of the concepts that we went over in uh, the earlier set of classes, and in this one, of course, we will talk about docking, the related concepts of that, the different algorithms that Professor Gumia kind of went over, etc., etc. Right? So we'll just get into all of this. So let's start the class with one question. So if I ask in drug design, what is the role of computational biology among these four options that are given? Which one do you think would be a more suitable option? So could you write maybe the answer in the chat, and then we'll look into it. So in drug design, what is the role of computational biology? Okay, so I see some answer B in the chat. So yeah, in this particular case, it should be to identify potential lead molecules, right? And now, if you look at the first option, it says identify drugs. However, drug is kind of like the final version of a study, right? Which takes a lot of years, and it's not just that the computational biology is itself enough to give us a proper drug. Of course, it can narrow down the list of molecules which need to be studied further in vitro, in vivo, etc. Then preclinical and then clinical trial and so on. But in the computational biology, while it can narrow down the list, ultimately we have to proceed for the wet lab experiments and the clinical trials and so on to get us a proper drug candidate. So you need to know that it has actually proper binding affinity, which is also verified, let's say in vitro processes, right? And then in mouse models and humans also this works without causing too much adverse effect. So all of this need to be done before we can call it a proper drug. So identified drugs should not be the proper answer here. However, for option B, identify potential lead molecules. So these lead molecules are basically the candidates which can be further proceeded for other studies, right? In terms of in vitro and in vivo studies, that can be obtained via computational biology. We know that in computational biology, through virtual screening or docking processes, we can basically overall estimate the binding affinity and binding interactions of, let's say, proteins with small molecules or ligands, right? And then whatever hit we get from that, we can go for some prediction of physicochemical parameters, and then we can proceed for a more computationally expensive and a bit more accurate processes such as molecular dynamics simulation, where we can simulate the dynamic behavior of the protein ligand complex inside a physiological system, or similar to inside a system that is similar to a physiological system. And then we can proceed for some rigorous free energy calculations from the trajectory that you got, where we will be able to say that, okay, this ligand is basically binding to the protein with a good enough affinity, right? So that can be done through computational biology processes overall. And through this process, the ultimate candidates that we will get, which can bind to the protein molecule with a good enough affinity, we can call them lead molecules. And after that, of course, we can proceed for further wet lab and in vivo studies and so on. Now, the C and D options, it says measuring IC50 values and measuring toxicity. Now, since the term they have used here is measure and not predict, so we should avoid these options for computational contexts, because measuring requires wet lab experiments, right? In computational processes, for example, using different web servers and so on, we saw earlier that they can predict toxicity. So they have already some trained model that have been basically trained on already available data. Uh, and through that model, we can predict the toxicity values of a new small molecule, which was not trained, let's say, for that web server in that model, right? We can get the prediction for these new compounds. And similarly, we can probably get the prediction for IC50 values also through some trained models. However, those will be prediction. Those are not measurement, exact measurement. For that, we need to go through wet lab experiments, right? So in this particular case, of course, option B would be a more suitable option. So yeah, that was a question. Now, in while talking about the binding of protein ligands, docking, etc. So one very related concept always comes in this context, which is QSAR, right? So can somebody mention what is the full form of QSAR? Firstly, what does QSAR indicate? So several QSAR concepts also were discussed by Professor Nomiha in week 10's content. So can anybody mention in the chat what is the full form of QSAR? So QSAR basically stands for quantitative structure activity relationship. So it's trying to basically find a relationship between the structure and the activity. And depending on what kind of parameter you are actually including in the study, they can be classified under 1D, 2D, 3D, 4D, etc. Different dimensions of QSAR, let's say, right? Now, in this case, I see one that is option D. I think someone has given an answer in the chat. So I should have been clarified that what is the unique about 3D QSR with this question. So 3D QSR basically what what basically differentiates it from, let's say, 1D and 2D. That is the surrounding atoms in the structure. So we'll just take a look about this question. Is kind of confusing in some sense, because of course molecular properties, connectivity indices, etc. can also be part of 3D QSR. Basically, whatever happened in 1D QSR and 2D QSR can also be considered as a part of 3D QSR, because it is not exclusive to the surrounding atoms in the structure. So I should have phrased this question kind of properly. But anyway, so if you look at the QSR, different types of QSR. So firstly, the 1D QSR, it basically correlates activity with some global molecular properties, for example, pKa, log P, etc. So different types of global properties, it just correlates the activity with, and that's how it gets the structure activity relationship there. Now in 2D QSR, so it correlates the activity with structural patterns, for example, connectivity indices, etc. Right? 2D QSR does not take into account the 3D representation. So only the connectivity indices, for example, which residues are connected to which one, and so on and so forth. So these type of kind of network or connectivity is considered by 2D QSR. However, the proper 3D representation is not taken into account. For 3D QSR, however, it correlates activity with interaction fields surrounding the molecule. So interaction fields means now the surrounding atoms or molecules, they also come into the picture. And in this particular case, we need the proper 3D representation of the molecules of the proteins to get the idea about the structure activity relationship. So 3D QSR is what the earlier question was talking about, and what basically what was unique about it was that it takes into account the surrounding molecules and the interaction fields and so on, which is you know happening because we take into consideration the 3D structure also in the 3D QSR while studying the 3D QSR. Now for 4D QSR, it includes ligand configurational ensemble. So ensemble is basically a group of different configurations that the ligand can adopt, and in the 4D QSR, all types of different conformations are taken into account, basically analyzing the overall ensemble to get the proper structure activity relationships and so on. Again, if we go another dimension higher, then in 5D QSR, we also take into account induced fit models. So in induced fit models, of course, there needs to be flexibility in the residues, right? In the protein residues and in the ligands and so on. So in 5D QSR, we have that. Through induced fit model, we can achieve that, right? And then in 6D QSR, now it incorporates even solvation models. Now, solvation models are basically representation of water around our system, right? So we know that in our physiological systems, of course, all the proteins, ligands, whatever protein ligand bound complexes, those will be in a solvated state, right? So to replicate that, we need to incorporate some sort of representation of water, which are done through different solvation models. For example, if we talk about some explicit solvation models, so solvation models can be explicit or implicit. Basically, in implicit ones, the entire solvation, basically the entire water phase, that is basically considered as a kind of a continuum. In explicit water models, the water molecules are defined explicitly. For example, there are TIP3P water model, which is widely used in simulations. So TIP3P is basically a 3-point water model where the oxygen and the hydrogens are only considered. More accurate models such as TIP4P are also available, which is 4-point water model. So additional lone pairs, etc., are also accounted for. So these are basically different solvation models that are also used in different molecular dynamic simulations and so on. And it is a part of 60 QSR in a way. So I see one hand in the chat. Yeah, please.
Sir, when we do docking, that time water molecules are deleted many
Yes,
times.
yes, yes.
Unnecessary atoms and water molecules are deleted. So, which stage this solvation models comes into picture, sir? Can you please explain?
Yeah, so in QSR model, basically, so in docking processes, I see that in different softwares, they will give you some options that if you want to delete the water molecules and so on from the structure before the docking, right? So that is good.
Yes.
That is what a lot of people do during docking processes. However, in solvation model, so QSR model is kind of different because you have to build the models and so on differently. Uh, and solvation model is particularly needed, if I say, uh, due to the fact that, of course, the role of solvent is also present while the protein binds to the ligand. Because a lot of times you will see uh, that the ligand will form, for example, water-mediated hydrogen bonds with the proteins and so on. Then there is also polar solvation penalties for the protein and ligand binding. So these sort of systems are basically a staple while we study molecular dynamics simulations, because you need to simulate the entire thing in a solvated system, right? But that's a dynamic process. In a QSR model, you can also take into account these type of solvation models where the role of water will be present there basically. So that
Okay.
will be a
So,
more computationally
sir,
expensive model. Yeah,
like,
yeah. Hmm.
sir, like you mean to say when the docking is facilitated by water, that time
No.
solvation model and 6DQSAR comes into play. When
So
docking
yeah,
itself is facilitated by water molecules.
so docking itself is so basically docking is kind of different. So that is in that case we are just trying to find the binding affinity. In QSR you are
Yes.
actually building a model for the structure activity relationships, right? And in that sense, if you include the solvation models there, so the different parameters, for example, the impact of the impact of water molecules that are surrounding the binding pocket, etc., uh, that will come into picture in protein ligand basically binding calculations. So that is kind of
Okay.
different there. With including solvation models, what happens is that you can get a more accurate result, because anyway your protein
Okay.
and ligand will be inside body in a solvated system. So that's just the entire point actually. But in
Okay.
a lot of papers, you will see that they generally stick to the 3D QSR model because it is, it does not have the highest accuracy, but it also balances the computational time with everything else. So takes
Okay.
lot, takes a lot less time, but it has decent accuracy. So people
Okay.
generally stick to 3D QSR in that sense. Yeah.
Okay.
Yes.
Thank you, sir.
Yeah. Yeah. Because as you will take further steps, for example, towards 4D, 5D, etc., that will also significantly increase the computational time if you don't even consider the solvation models, because configuration ensemble means that for every configuration that you can find for the ligand, you have to calculate the interactions and so on, right? You have to build the model while taking into account all of these. Same with induced fit models and so on. So these are quite highly computationally expensive. So a lot of people you will see they will just stick to 3D QSR models because it is kind of a balance between high accuracy and balance between basically decent accuracy and good computational time. So these were basically included in Professor Gumia's slides also. So I just took the reference from that. Now let's say we just switch to the other process that is quite widely used in binding free energy predictions and so on. So that is docking, right? So in protein ligand docking, what are the two important bottlenecks? Uh, so four options are given here. So could you try to maybe answer this in the chat? Classic. So when Professor Gomiha introduced docking, he talked about the two most important elements that are present in docking, right? So one has to do with uh, the ligand geometry kind of inside the binding pocket, and the other has to do with actually calculating the binding affinity or the binding energy, right? So the two most important bottlenecks for protein ligand docking, what do you think those are? So I see one answer D in the chat. So maybe others can also try to answer. So in some of the further classes, we will also see examples of protein ligand docking through the use of some softwares, for example, Autodoc Vina, or something like that. But while talking about docking, these two important elements or bottlenecks are quite useful to know basically. I see also A and C. So in this particular case, binding site prediction is definitely important, but that is usually not the part of protein ligand docking. So here, the two important elements or the two important bottlenecks that are kind of meant as. So these are conformational sampling and scoring function. So when Professor Grumiha introduced docking, of course you have to do the binding site prediction beforehand, or you can do a blind docking kind of thing, right? But in the actual docking process, so in the actual process of finding the perfect geometry as well as the associated binding free energy for that geometry, so that's when the conformational sampling and scoring function come into picture. So the two primary elements are docking are, of course, one is search algorithm. So the search algorithm is basically the process via which we find the particular pose for a ligand in a binding site. Okay. So what it does is that it generates a lot of poses of a molecule within the binding site. So these poses are basically different geometries of the ligand that the ligand can be present in inside the binding pocket. And it basically involves an extensive process where the possible configurations that the ligand can have inside the pocket are explored. Now, it can be explored through a more rigorous process, or we can take some uh, basically leniency there. So we'll talk about that maybe. So scoring function is also the other component of docking. So what the search algorithm is doing is that it is finding the particular pose or the particular geometry of a ligand inside a binding pocket, right? And this is what kind of the conformational sampling part was talking about, because different poses are different conformations, right? So conformational sampling means you have to sample through all of these conformations and try to find the binding free energy corresponding to these conformations, and then you can get the best pose and their corresponding binding free energy, right? So that is the conformational sampling, which is done by the search algorithm. The other component is scoring function. For each pose, the scoring function determines the score or the binding affinity between the ligand and the protein. So as it is written here, it evaluates the strength of binding between the molecule and the site. And these two components overall are the crucial parts that any docking protocol will have. Now, while trying to find out the proper search space, the search space will include, it basically means searching through the different conformations and trying to identify which conformation is possible for the ligand inside this pocket and so on. So while searching for these conformations, uh, there come certain challenges, because of course, since your molecules will have multiple degrees of freedom, that means translational, rotational degrees of freedom, as well as different conformational shifts. Further, of course, if you have solvent molecules present, then they can also influence ligand protein interactions, which overall adds to the complexity. So that is why, due to this large number of degrees of freedom, if you try to basically go through every single conformation and every single basically combinations possible, so it will take a lot of time. It will be vastly computationally expensive, and it will take a large amount of time, right? So what the search algorithms try to do, so they try to explore the search space, but try to do it in a bit more efficient way, basically where the balance and the coverage of the conformation space is kind of considered together. So these two, the time and the coverage, these two are kind of taken in a balance, right? So those are the challenges in the search space. Now, while we're talking about docking, there are overall kind of two types of docking that involves basically restricting the movement of the particular molecules and the protein residues or not. So depending on that, we have rigid versus flexible docking. So what rigid docking is basically, it treats the protein ligand they as rigid structures during docking, so they cannot move around. And it only considers the translational and rotational degrees of freedom. However, on the other hand, flexible docking, they incorporate conformational changes and they allow for a larger search space, uh, but require more computational resources, because when you have the search space that large, then it will take a lot more time and it will take higher computing powers. Now, depending on whether you have predicted the binding pocket or not, sometimes people also categorize docking into, let's say, blind docking, or just in a predicted binding site docking in that region. When a blind docking, a lot of people, what they do is that if they do not know the pocket, so they will just generate a grid and they will cover the entire protein with it, and they will try to analyze all of the different basically cavities that the ligand will go and bind, and they will analyze all of these different protein ligand bound complexes separately, kind of, after the docking process is done.
Excuse me. Excuse me, sir.
Yes.
Sir, in the previous slide you mentioned about flexible this thing, and you also told that this needs high computational resources. So does that mean I cannot do this on my PC or desktop?
No, no, no. So it depends on basically how many compounds you are trying to dock, right? So nowadays, of course, modern PCs, desktop or modern laptops, they are quite, they have the power to perform like simple tasks like docking, right? And you can do that for, let's say, 10, 20 molecules, one after another in your computer. You can go through that. You can do that in a flexible way. Uh, so if you use Autodoc, for example, Autodoc Vina or something like that, then you will be given the choice of which residues do you want to consider flexible and so on, right? Which bonds do you want to consider rigid and these kind of things. So you can do it in your laptop, but let's say for, let's say, certain number of molecules against a particular protein, but let's say you are trying to screen a large library. Okay, so large library means, let's say it has lakhs of molecules. Now in that case, the more uh, flexibility that you consider in case of your proteins and ligands, so the more computing power you will need, basically the more time it will take. So in that sense, you might get limited in some way, because if you have, let's say, 10 lakh molecules to dock, so it will take forever if you consider a lot of residues flexible in the binding pocket, right? So that is what is the main concern when we talk about uh, the flexible docking, because it can take a lot more time than if you consider it rigid. But if you have limited number of molecules, so small number, let's say 10, 20 molecules, then you can do it one after another. That's fine. If you have good GPUs in a desktop system, then you can also go for a flexible docking with a large library. So that is also possible. And if the particular protocol allows you to accelerate the docking processes through GPUs, then that's even better. So that is also fine. You can do it also in that way.
Okay, so otherwise, if I have a larger library, so maybe some computer labs or there I can start the program, and
Yeah.
means like it will be screening the lakhs of molecules. So it will not be like some 10, 20 at a time, then next 20 or maybe some 50 molecules at a time, and like you said, it will go infinite. So can't I means like if I have already screened some 50 molecules or 100 molecules, I can, I mean, maybe in weeks time or a month's time.
Yeah. You can do that. You can do that definitely. So for screening of large libraries, so some softwares, some commercial softwares, for example, Schrodinger. So what they do is that they kind of take a sequential kind of approach. So firstly, they will screen all of the molecules in a lower accuracy algorithm in a high throughput way, and then they will proceed whatever hits they have got from this initial stage of docking. Then they will proceed with a, let's say, little better, a little more accurate algorithm, and then whatever hits they have got, then they will even proceed with a the most basically extensive docking algorithm that they have. So kind of in a sequential way. So they first weed out all of the unwanted small molecules in this high throughput part, and then after that again in the sequential in the next step, basically they take a bit more accuracy algorithm, and then they again take certain hits from that and then go for their highest accuracy algorithm. So some softwares have these kind of protocols. Others, they just follow their highest accuracy algorithm for all of the compounds and so on. And also, I think in different docking softwares, you will get the option to basically choose what kind of flexibility you want. So, for example, mostly ligands are kept flexible nowadays. So instead of everything is like rigid, so the ligand molecules are kept flexible, which will, or let's say most of the bonds that are present in the ligands, those are kept flexible. So their rotation, etc., that is possible. And the other protein parts, let's say, they are kept rigid. So that is also kind of a balance that you can take. So a lot of people do that kind of thing also. So it just depends on what kind of performance you have in your computer and what kind of in what time you want to get the results and so on. And also it depend on the size of your library, of course.
Okay, thank you.
Okay. Yeah. So types of search and flexible docking. So search means, of course, finding out the different conformations and going through the different conformations, basically, right? So one, we have the systematic search. Now it is an exhaustive approach. Because it is exhaustive, it tries to exhaust all of the options that can be possible, right? So it is more computationally expensive, takes a lot more time, and only feasible for low dimensional problems with limited degrees of freedom. So let's say if you have a very large protein, uh, so then again, because of the number of atoms in this large protein is very high, so your dimensions of the system will increase, right? So for that case, you will need to consider a lot more degrees of freedom, of course. And if you try to do kind of an exhaustive search process, it will take a lot more time again. So in that case, what people do is that they kind of rely on a stochastic process. So stochastics basically it kind of utilizes a randomness to explore the search space. So it creates random conformations kind of, and then tries to compare these different conformations, and then they kind of take a step from there. So if it finds one conformation that is particularly good, then it will take let's say slightly modify this conformation and then uh, try to consider the other conformations that are kind of related or kind of like close to this kind of the initial hit that they got basically. So for example, simulated annealing genetic algorithm, taboo search, these are some examples of the stochastic search algorithms. And for higher dimensional problems where the exhaustive search approach will take a lot more time, so for that these kind of algorithms are basically used. And of course, you can do a global search with a local refinement approach, which will kind of be a hybrid approach. Some softwares, for example, Autodoc or Lamarckian genetic algorithm. So they are the examples that include the hybrid kind of process, right? So if we talk about, I think we'll talk about some of these algorithms maybe in some later slides or in later classes. But if we, let's say, try to answer this question, so this relates to the steps that you have to take in order to get a QSR model, kind of, right? So in QSR processes, what will be the correct order of steps that you have to follow? And 5 options are given here. So which of them could represent the correct order? So could you please try to answer in the chat, and then we'll look at it. Follow the steps one after another, make, while constructing the QSR models. So which steps do we need to follow follow first, and then what could be the next step and the next step and so on? I see C option in the chat. Yeah, we'll just look into it. So the first step that we need to consider for a QSR model is, of course, since it relates to the structure activity relationship, we'll first have to uh, go through the already available data that is present, based on which we will try to construct the model set. So first step in this process, it will be the option 3, as it is mentioned here. So collection of ligands with experimentally known activity. So the experimentally known activity will data we need to collect, and then following that, we need to generate the descriptors. Now these descriptors will be kind of the parameters in a way that will define the ligands, basically the structure of the ligand, the geometry and of the ligand, and so on. So those will be classified as descriptors or categorized as descriptors, different descriptors, right? So after we generate these descriptors, then we have to do the feature selection. So what feature we want to include in this particular model, and then we have to construct the model, and of course finally we have to validate this model, because without validation we cannot say for certain that that this model is good, right? So in the right hand side figure, we also see that of course dataset preparation and then molecular descriptor generation, and of course there is some training set, test set, and so on. So in training set we will do model training, and after we get the final model, then we have to do the validation and so on, right? So you might have also seen like similar type of approach in a machine learning kind of algorithm, where there you also need to do these training set, test set, and ultimately validate the models. But yeah, in this note on the right hand side, you can see that in the particular data set that you want to use for generating the QSR models, so they need to have adequate number of compounds with wide activity range and consistent biological activity data. So adequate number of compounds are needed because you want to get statistical significance. Wide activity range will allow you to basically classify that, okay, this type of ligand is associated with this type of activity. Similarly, this type of ligand is associated with this type of activity, and so on. So from a low activity range to a high activity range, all of these different values of activity, let's say, can get associated with a particular type of ligand if you have a wide activity range in your data set. And then the biological activity data, they need to be consistent, of course. And then after that, you have to compute or measure experimentally a number of molecular descriptors. So these descriptors will reflect the properties of the compound. So it will be the physicochemical properties of the compound for each compound in the data set, right? So let's say some simple descriptors could be, let's say, molecular weight, the volume of the ligand, the number of rotatable bonds that are present in the ligand, the number of rings that are present in the ligand, so on and so forth, right? So these different types of descriptors that basically describe your molecule in an efficient way. So they need to be selected, and you have to consider which descriptors you want to use for your model. Then, of course, you have to build the training set, and then predictive ability of the model. To examine that, you have to also build a test set. Then after that, you will just derive the quantitative correlation using some statistical methods. And then also in that process, you have to consider which statistical model do you want to use. And then internal validation in the training phase and external validation in the predictive power model for the test set, they need to be done. So while constructing the model, the first step is collecting the activity data for already uh, basically reported ligands, kind of, right? So the ligands that have already been identified through experimental processes to have this activity value. So you need to first take that into consideration. Now, there are some databases which have this sort of collective data for experimental activity of ligand against certain proteins. So any idea? So do you know any example of this type of databases which have the experimental activity data for proteins and ligands?
PDB database.
For PDB database, you get the crystallized structure of protein ligands, but they do not always have the activity data, right? So the experimental activity data, one widely popular database is this one. So this is bindingdb.org or binding database, as you can see here. So this is a very good database, and for a lot of proteins, whatever experimental data regarding ligand binding and so on that have been reported, that is kind of cataloged here basically. So here you can go to these, let's say, different options, and you can try to identify the protein and the respective ligands that they have the data for. So you can search for the different targets, for example, through names. So if we just, let's say, randomly,
Sir, kindly highlight the search bar like at
just
ID.
yeah, just wait a minute. So in this search bar on the initial page only, I will just try to get the search done.
No, sir, the address of it. How
Uh,
to type in this, get this software, I mean, this
ah,
site.
okay, okay. So you just open a new tab.
Hmm?
You search binding DB. Okay. So this binding DB database is basically the name of the database. And if you just press binding DB on Google Chrome or any other browser, you will get the bindingdb.org, www.bindingdb.org, this link. So it should come as the first link here. And then you just press that, and then you will get the actual database, get to the actual database. So here they have a lot of options basically, targets, compounds, publications, special datasets, and so on. But let's say we just want to find out the data regarding a particular protein. So as what I did in this tab here, so I searched for splint tyrosine kinase. So this is a protein molecule, of course. And while searching that, I just got a search result here on the top. So if we just open that up really quickly, so it may take some time, but we'll notice that against this particular protein, a lot of data, experimental data, whether they would be Ki, they would be Kd, the dissociation constant, or the IC50 values and so on. So those will be reported, right? And they have the options to download the particular results that you are getting as datasets also. So that also can come in quite handy if you are trying to build a QSR model, or conversely, if you are trying to build a machine learning model to predict the binding of new compounds, right? So yeah, so the splint tyrosine kinase search, through that, we are getting these sort of different results. So we'll see that it seems 7536 total results have been provided by this database. Of course, in the first page, we'll only see like 50. Then you have to again go through manually the next pages and so on. But if you focus on the particular data that it is providing, we'll see first of all on the left hand side, we have the target name, the source, maybe whichever company have reported this data, and then the particular ligand that these binding activities kind of being calculated for. Their smiles also. Then we have the affinity data. So in this particular case, it is talking about Kd, right? The dissociation constant, which is in nanomolar, then 600 nanomolar for this one, for example. Then what kind of assay was performed to get the data, then the target info, ligand info, and then some other stuff is also given here. So we can search for, let's say, different types of values. If you want to particularly see those, for example, if I want to see Kd, then I could just search for whichever values are reported for Kd. If I want to get the Ki data, the inhibition constant, then I could get the result for Ki. I think here some search issues are happening, but anyway, excuse me. So you get the idea. So you can get the different values from here. You can also, I think, press this make dataset option, and then it will, let's say, add all pages, and then you can make the dataset. So basically, you can download this dataset after verifying. So they are providing, I think, a 2D SDF file, and then also 3D structure they are providing these type of things, and also tab delimited. So this will kind of be like a like an Excel file, right? So they will have the data there also. So you can download this kind of data, and whatever is needed for your particular research, you can get that from here. So it is quite useful website. I think Professor Bhumiha also talked about binding DB in some of his lectures. So if you need to make some sort of models and you need experimental data regarding binding affinity, you can refer to this particular website. Okay. So these type of available databases are quite useful if you also want to build some machine learning models, right? Because with experimental binding affinity, you can maybe get good accuracy for your models also. So another approach that some people have used to reduce the computational time and so on, that they have went for let's say broad deep learning approach, where they will just talk some part of the database through a talking protocol and then use that to train their model. So that is another approach that you can do if you do not have the experimental data available to train, let's say, against a particular protein. It's a new protein, it has not been worked on that much, and you want to predict some molecules for this. You want to build some model for this prediction, right? So what you can do is you can take a large database and not use all of the data that is present in the database, but use certain only like a small part of the database, and you can train a machine learning model through that via getting the docking scores for this small part of the database, and then you can use this trained model to predict the scores for the rest of the molecule that are present in the data. So we'll see that maybe in some slides. So yeah, so while talking about scoring functions, which is basically the uh, part of docking that will give you the value for the binding affinity or the binding free energy, right? So different kind of scoring functions are available. So for example, one of them is force field based scoring function. So Professor Bromiha went over some examples for these different types of scoring functions, and basically some widely used softwares that use those. So could you maybe try to answer this one? So Autodoc, we know it is a docking tool. ChemScore is also a software available. Glide, that is the part of the Schrodinger suite, and PMF is basically potential of mean cells. Okay. Yeah, so as we see here, so in this particular case, a force field-based scoring function, one example for that will be the Autodoc, widely used Autodoc software. And other than Autodoc, Gold Score, etc., these are also force field-based. When you talk about empirical scoring functions, so there comes ChemScore, PLP, Glide, SP or XP. So what I talked about in some of the other slides, that some softwares basically allow, let's say, different sequential approach in docking, right? So first they will go in a high throughput way, then the heat from the high throughput approach, they will proceed through the different, let's say, slightly more accurate docking protocol and so on, right? So one example of that was Glide, which I was kind of referring to. So the Schrodinger Glide, they have three different consecutive basically docking protocols. So the first one is HTTPS, as they call it. It is a high throughput virtual screening, useful for screening a large number of libraries. And then after that, you can proceed. You can basically take the hits from the HTTPS through this SP. So this SP is kind of a standard protocol docking. So it balances time and accuracy. And then after that, for example, when you have reduced the number of hit molecules by a lot, then you can proceed for a XP docking. So XP is the most extensive kind of protocol that they have, docking protocol. So these are kind of examples of empirical scoring functions that they use. And then in case of knowledge-based potential, so we have potential of mean force, drug score, ASP, these different types of approaches they use. So I think this information was included in Professor Bhumiha's slide. So you can take a look at that maybe to know some other related stuff from these kind of different scoring functions. Okay. Now, if you talk about the empirical scoring function, which we looked at in the other slide, so it basically involves calibrating score parameters to mimic experimental binding affinities. There are certain advantages. For example, it has a rapid estimation of binding affinity directly from the model, but also there are some disadvantages. For example, complexes with accurate structure need to be available. So if there is limited availability for these kind of complexes with accurate structures and the binding energies that are known, so that will put the scoring, empirical scoring kind of in a bad place. Now, if there are measured binding affinity inconsistencies for certain protein and the certain ligand, then that will also create a disadvantage. And it basically relies on the placement of hydrogen atoms. The transferability is influenced by the composition of training set, how you do it. There is also effective penalty lacking for inaccurate or poor quality structures. So this is kind of an overall formula for the empirical scoring. We will see that the different energy parameters are given here. For example, energy parameter corresponding to torsions, hydrogen bonding, ionic, aromatic, hydrophobic, etc., and so on. Right. So this type of thing, this type of kind of a overall consideration of different energy parameters are used in empirical scoring. If you talk about the force field based parameters, so what force field are basically? So it is a mathematical model via which you can calculate the potential energy of a system's atoms based on their coordinates. Okay. Now you can optimize these parameters in empirical calculations also, and then force field based parameters, they can reach accuracy comparable to quantum mechanical models sometimes. So basically, since it categorizes the system's free energy, that is a dependable algorithm for exploring the conformation space. And we know that in molecular dynamics simulations, in classical mechanics also, and so on, the force field are used for calculating the potential energy, right? So the different types of force field are used to basically characterize different types of molecules, biomolecular molecules, and so on. For example, in proteins case, you will know that, let's say AMBER, they use different force field. Let's say FF14SP or its updated version FF19SP. So these are different types of force field that AMBER uses for the AMBER software suite uses for categorizing the proteins. So you need to load this force field and their information first to get the protein to be loaded properly, essentially in a system. And then, for example, in case of the different phosphodilated residues, so it's a phosphoserine, phosphotyrosine, and so on, then you need to load another force field because the original FF14SP and so on, these are not suited for them. So they do not have information for the additional atoms that that are contained in this phosphodilated residue. So you need to load another force field like phos-AA10. If you want to characterize small molecules, then there is another force field for that, which is called the generalized AMBER force field or GAF. So these type of different force field are used to basically effectively kind of characterize your particular molecules, right? Now, if you talk about the different, yeah, I see one hand in the chat. Yeah, please.
Sir, like I have used the tool Clustal Pro.
Cluster Pro.
Clustal Pro.
Yeah.
Yes, Clus Pro from Boston University.
Yeah.
Like they usually dock like whatever we give as ligand and protein, they dock with that. So like which category this Clustal Pro belongs to with respect to scoring, like force field based or force field like
So,
that? Anything can be categorized like that?
yeah, definitely it should be categorized like that. But on the top of my head, I do not know what Cluster actually uses. Let's see. So here they are talking about, I think they have something called Piper scoring function for
Yes.
class pro. So I do not exactly know what category it should belong to, but maybe I have to look into it. But yeah, they
Sir,
will
like
be categorized in one of these one of these approaches, or it could be a hybrid kind of approach containing multiple different type of parameters. Yeah.
Sir, one more thing, as you were talking about the targets, ligands with respect to the proteins,
Yeah.
which ligand will bind to which protein. But as I have seen in classes, whenever we provide any ligand and any protein, they do dock,
Yeah. Yeah.
but scoring function is different. Of course, negative, more negative means it's stronger affinity, and less towards positive means it's less affinity. That scoring function is given,
Yeah.
but
The
they
scoring
do
is given.
dock.
Yeah. Yeah. They do doc.
Yes.
So yeah. So that will always kind of occur. So the algorithm will try to see the best kind of geometry and the corresponding best score that it can obtain. Now, if it is, let's say, a very bad value, for example, it is -1 kcal per mole, something like that, then it is like
Mhm.
very loose binding. So you cannot consider that probably as a effective small molecule against that protein. But it will always generate something, whether it will be loose binding or
Yes.
a good binding or something like that. So it will try,
Mm.
it will do the best that it can do for this protein and this ligand. So that
Yes,
always
sir.
happens. That's why since also you have uh, the basically the docking processes are always not that reliable because they have lower accuracy. They are kind of the dynamic nature is not considered. So that is why you probably have to go for a higher kind of set of study in that sense. So a more
So,
computationally
like
efficient.
Autodoc Vina is the highest accuracy, means
No,
attainable accuracy.
no, no, no. It's
Autodoc Vina.
no, no. It's not like that. So I'm talking about just the protocol of docking for any software that you use. Since, for example, in most cases, the protein will be considered rigid, right? Protein will be considered rigid, and then the ligand will probably be some dynamic. You can put some bond restraints in there and so on. Uh, but since the actual nature of the binding of protein ligands are not, let's say, in this rigid space, and then the ligand is kind of changing the conformation, it's not like that. So everything is dynamic. So that's why I was talking about that for your lead molecules or hits, whatever you get, you have to kind of proceed for a more dynamic study, for example, like MD, molecular dynamics, and so on. So that's what I was getting at. But for docking protocols, docking approaches, all of the softwares that are generally used or are highly cited by different studies, so they can be reliable basically. You can think of them as reliable. But if you just want to focus on docking, one thing you could do is that, uh, for example, the set of molecules that you are docking, so you can dock them through multiple different servers. For example, you use Autodock, let's say you use ClassPro, let's say you use some other software or other servers, so let's say Schrodinger you use. So these different types of softwares, if you dock the same set of molecules against the same target receptor, so then you will be able to compare and contrast. Okay, so if we see similar kind of patterns, then that is probably going to be reliable, something like that. Okay, so to validate, you can try to do that in your docking processes and so on.
Okay, sir. Thank you, sir.
Hmm. So here we see that the force fields can have different types of uh, basically categories. For example, all atom, united atom, or coarse grained. Now, when you talk about coarse grained, it is basically taking not considering each and every atom of these molecules as a individual unit, but it is considering a whole sort of, let's say, residue as a particular bead, something like that, right? So it is, of course, going to be lower accuracy, but uh, you can simulate a much larger system, or you can describe a much larger system using coarse grained force fields, because since you are not considering each individual atom as its separate unit, uh, so the coarse grained models will uh, have uh, basically lower computational time as compared to your all atom models and so on. So coarse grained will be useful for if you are wanting to simulate a much larger system, kind of, let's say you have a lipid bilayer and then a lot of proteins are bound in this lipid bilayer, a lot of transmembrane stuff is there also. So in that kind of sense, since the atom number would be much, much higher if you consider each atom separately, so you can go for something like coarse grained model. Uh, Correspondingly, conversely, if you consider these all atom models, for example, So in all atom models, every single atom will be its individual unit. Like every single atom needs to be described separately. So in that sense, of course, the computational cost will be much higher. The computational time will be much higher. But for focusing on certain small proteins, let's say it is like 1,000 residue or 300, 400 residue proteins. So for that, those kind of systems, you can go for all atom based force field, because you can get good accuracy with that. So good description of your particular system with that. Now for the molecular mechanics, we see that the total potential energy, they have bonded terms uh, and also non-bonded terms. So bonded means covalently bonded atoms, and the non-bonded means the interactions such as van der Waals, electrostatic interactions, and so on. Right. So these are all part of the force field. The non-bonded potentials, the bonded potentials, and so on. These are part of the force field. Okay. So what are the excuse me. So what are the advantages of force field based scoring? So it is well researched in terms of physical foundation, because you are strictly using the potential calculations, you are using the Newton's laws and so on. So these are basically vastly researched, and they have been improved over and over and over, so that the modern generation force fields, let's say, which are widely cited and widely used in different studies, so they are pretty reliable, you can say, in terms of their the physics foundations, right? And they are transparent and efficient when applied to a pre-calculated grid. Now, there are certain disadvantages. For example, sometimes they tend to overestimate electrostatic interactions, uh, and then they only contain certain energy aspects like potential energies, occasionally complemented by solvation and entropy terms. So these are some limitations or disadvantages, but overall force field based uh, scoring is quite good. Can give you good results in terms of docking, or if you use in molecular mechanics for MD simulations also. So these will also provide good results. So in terms of the different potentials, for example, in force fields, so you see that V total can be classified into V bonds, so potential for bonds, then angles, dihedrals, the improper, and then van der Waals, electrostatic, and so on. So in the bonded potential, it will kind of look like this. In the angles, of course, these are, or let's say the coordinate vector, that is kind of represented or replaced by the angle terms. For dihedral, again, the formula is kind of given here. Uh, Something like van der Waals and electrostatic interaction, we know that electrostatic is calculated using the Coulomb formula. So you need the charge and the distance between the charges to apply this formula. And then the van der Waals, this is usually calculated using something like Lennard-Jones potential for the van der Waals energy parameters. Now the other one was knowledge-based scoring function. So what it does is that it utilizes free energy of molecular interactions, which can be obtained from structural data from protein ligand complexes from databases like PDB. And it applies Boltzmann-like statistics to analyze interatomic contacts. Now, what Boltzmann-like statistics is that I think we went over some of the examples in some other classes, where basically based on some parameters, let's say population difference, something like that, you can calculate the energy of certain states, let's say. For example, when we talked about, I think in some of the classes we talked about free energy landscapes in terms of different values, right? So what we do in that case is that we are calculating, for example, it's just a simple example. So we're calculating RMSD of a protein, right? Root mean square deviation, which can give us insight into the deviation from the initial conformation. So we're calculating that value, fine. Now what we can do is that based on all of the RMSD values that we have got for a single molecular trajectory, molecular dynamics trajectory, we can basically categorize the range of values that it has into separate bins. So bins are basically sub ranges for the basically if you add up all of these sub ranges one by one, you will get the entire range. However, within this particular small sub ranges or small basically ranges where a lot of values are basically obtained for this particular sub range inside these the small boxes, if you say. So we can, based on the population of the number of data points that we have for this sub range or for this box, we can calculate a relative energy value for the conformations that are present in this subrange. In terms of RMST, if you are considering RMST here, so you can do that. And this relative sort of energy will be basically computed against the most populated bin or the most populated subrange that we have. We'll compare the population of the other subrange with this particular one, and then we will compute the energy. So in that sense, the most populated bin, that will be the lowest energy or the minimum state, minimum energy state, and the other bins that we have all around this particular one. So they will have certain level of energy, right? So based on that, we will be able to see, okay, so this is a high energy state. And then this is the most probable state, which are the minima structures. So if we have multiple minimas scattered across different spaces, we can say, okay, these could be probably the intermediate states that that is present in this conformational ensemble. So these kind of things we can do, and that is basically an application of Boltzmann statistics, where you are using the populations to calculate the energy value. So in the knowledge-based scoring function also, for example, you can apply this Boltzmann-like statistics. And it has an advantage that it relies on a vast amount of distance data from structural information. So it makes it versatile compared to empirical methods. And it also incorporates structural knowledge, of course. But they had some disadvantages. For example, in Boltzmann hypothesis, there is an assumption that it is a spatially uniform liquid, which is not the case for protein ligand complexes. And they have also the pairwise limitations because you need to calculate the forces basically pairwise, kind of like that. So they have those disadvantages. So this is one formula example that I was talking about in terms of Boltzmann kind of energy value formula. So this delta G equals minus RT ln of something, right? So in this case, we have ln of propensity. So if you wanted to compute the relative energy in the earlier cases, it will be ln by n divided by n0. So the relative basically fraction of population in different bins compared to the most populated one, right? So in this particular one, they have propensity value. So these different kind of parameters also you can use as your input, and then you can get the energy values, and you can draw a landscape of the possible, basically the possible space that you are covering there. So that is kind of the overall basis of knowledge-based function. So again, these are also included in Professor Brumia's slide, so you can take a look at that also. So you just, I think, looked into it. So what is the type of algorithm used for genetic algorithm? So genetic algorithm, I'll just go through this probably very briefly. So it is one of the stochastic search algorithm. So stochastic search algorithm basically means that it utilizes randomness. So stochastic means randomness. So it utilizes a stochastic approach to generate the random conformations in case of genetic search algorithm, and it is also a good algorithm to get the conformational ensemble, right? Now, what it is that for solving global optimization problem, of course, it is relying on a stochastic approach. The steps for the algorithm is, firstly, you have to initialize the algorithm. Now, it will generate an initial population of chromosomes randomly. Now, here we are not talking about literal chromosomes in biological sense, but we are talking about certain, let's say, conformation for the molecules, right? So these, if we just use the term chromosome from for these particular conformations. So here what it is trying to say that it is generating those conformations randomly because it is a stochastic approach. And then after that, what it does is that, so we'll get into that. So here basically each chromosome is representing a particular pose. So that means that we have a diverse set of poses inside the binding site. So it's not literal chromosome, but it's particularly representing one conformation or one pose inside the binding pocket. Now, after that, so it pairs the high scoring chromosomes or the high scoring poses to produce children, right? So these are referred to as parents, which are kind of combined to produce the next set of poses. So that means that if, let's say, certain poses, let's say pose 1 and pose 4, they have good scores. Now in the next step, what the algorithm will try to do is that it will look at both the pose 1 and it will look at the pose 4 at the same time, and it will try to generate combinations of it as part of the next conformation that it wants to evaluate basically. So let's say it will take this particular ring orientation of this pose 1, and then it will take the side chain orientation of this pose 4, and then it will combine that and generate a new pose kind of thing, right? So due to that, basically, so it will combine the attributes of the high scoring poses to generate a new pose. And as I said, for example, location of one high scoring pose with the torsion angle of another high scoring pose to create a new pose. And basically, it will say that, okay, since we are combining the two high scoring poses, it could be possible that the new pose that we are generating, it will also have a high score. So this kind of uh, particular process via utilizing randomness and then combining the best scoring poses together to generate the new poses, so that uh, allows it to limit the search space by a lot, which is why it is quite like efficient in terms of identifying the proper binding poses for the ligands, so on, right? So then what it does is that it randomly mutates children to introduce variability. So the children or the new poses, which are basically combination of two previous poses, two previous good scoring poses. So it generates mutation into that to introduce variability. Basically, it makes some changes in this particular new pose, and it sees that, okay, whether this change kind of leads to a better score or conversely it leads to a worse score. So it kind of evaluates the new pose for that through that. For example, it can alter the torsion angles or orientations randomly in the child pose, and it will just try to see, okay, whether this new orientation or this new torsion angle for this particular uh, set of bonds, whether that leads to a better binding score or better docking score or not. So it will try to do that. And then ultimately it will do the selection of fittest. So it's basically from a current population, it will find the fittest individuals. So in the docking sense, it will have the best scores, those kind of poses. And then it will try to just generate the it will try to get newly generated poses to form the next generation. So it will again combine the best scoring poses and it will again form the next set of poses for the next generation. And it will kind of do that iterative process basically. So after iteration, let's say n number of iteration. So if you do not, let's say after a certain iteration, if you do not see any significant changes in the next iterations, then you can say that, okay, so the step has completed. And then the results that you are getting, we cannot get any better results than this. So we have kind of converged our algorithm in a sense, right? So it is saying that it will continue until a high scoring pose is identified or until the convergence criteria, whatever is set, they are met. So this type of way, genetic algorithm uses the randomness to generate poses, and it kind of evaluates them and creates new poses from the parent ones, and then tries to evaluate them, tries to alter certain parameters inside these poses, and then again tries to evaluate them, again tries to basically combine the two of the fittest uh, in the next generation and so on, and it will just keep doing that. So that is how the genetic algorithm kind of works. Okay. So here it is talking about chemical complementarity score. What does it mainly depend on? So could you try to maybe answer in the chat? So chemical complementarity will basically be whether one molecule, let's say one small molecule, can complement another target receptor, right? So they need to have, of course, geometric compatibility or geometric complementarity, and then there needs to be also formation of different kind of interactions that will stabilize their interactions, right? That will stabilize their binding. So in this cases, we need to consider both the hydrogen bond donors and also the hydrogen bond acceptors from the proteins and ligand side also. So here the interaction based question was kind of what they are getting at, and I see answer in the chat also. So basically the hydrogen bond donors from the ligand will try to form bonds with the acceptors of the protein, and conversely the hydrogen bond acceptors of the ligand, they will try to bond with the donors of the protein, right? So this kind of way they will get, they'll kind of complement the composition of each other in a sense. Okay. Now, if we talk about some numeric values, so here one question is being asked. So it says that the IC50 values of 3 protein ligand complexes are given as this, right? 6 nanomolar, 15 nanomolar, and then 2 nanomolar. Now, another complex you have where the IC50 values are related with the hydrogen bond donor. So in this particular one, uh, we have 3, 2, and 1 value for hydrogen bond donor. Then we have 5, 6, and 5 value for hydrogen bond acceptor. And then we have 4, 1, and 2 for polar surface area. So the first value of basically this brackets or this parenthesis will correspond to this first value of the protein ligand complex IC50, right? So for example, for 6 nanomolar, we will have 3 hydrogen bond donor, 5 hydrogen bond acceptor, and 4 polar surface area score. For 15 nanomolar, we have 2 hydrogen bond donor, 6 hydrogen bond acceptor, and 1 polar surface area score, right? So this type of way, they have given us some parameters which we can assume that are contributing to the IC50 value. Now, what they're asking is that the regression coefficient for hydrogen bond donor, what that will be. So for hydrogen bond donor in this particular case, we had 3, 2, and 1 corresponding to the 6, 15, and 2 nanomolar protein ligand complexes. Okay. So they're asking the coefficient for the hydrogen bond donor in that sense. So these type of numeric questions, you can solve it quite simply. So I'll just go through the process maybe. So what you have to do essentially is that you have to basically allocate a variable for each of these parameters. For example, if you allocate X variable for hydrogen bond donors, then Y for hydrogen bond acceptor, and Z for polar surface area, then what you can do is that whatever the values or the number of, for example, hydrogen bond donors and acceptors and so on that have been given, you can just multiply that with the particular these variables, these coefficients, and then we can just do a summation and expect to get the ultimate value for the protein ligand complex IC50. So for example, for the first protein ligand complex, we have 6 nanomolar value. So 6 is on the right. And how do we get this 6 value? We are asked to basically consider the 3 hydrogen bond donors, 5 hydrogen bond acceptors, and 4 polar surface area to get this value of 6 nanomolar. Right. So it will have 3x plus 5y plus 4z equals to 6. So this sort of equation you can basically put for the first protein ligand complex. Then again, for the second set of the second complex, basically you have 2 hydrogen bond donors, 6 acceptors, and 1 polar surface area. So you can just put like 2x plus 6y plus z equals 15. And then for the last one again, you can put x plus 5y plus 2z equals 2. So for 2 nanomolar, right? So you can just solve this equation, and then ultimately you should get the coefficient for hydrogen bond donor, which is in this particular case they are asking for the value of x here. Okay, so that's a numeric question. What you can do basically is that, let's say from equation 1 you can get the x value kind of like this, right? 6 minus 5y minus 4z divided by 3. Then from the equation 2, overall you can get something like this, y equals 5z plus 33 divided by 8. Then you can put the value of x in equation 3, and if you try to solve it, then you will get the value of z equals to minus 5 ultimately. And then you can put, let's say if you get ultimately one value, you can put that in the other equation, try to reduce its complexity, and then you can combine with other equations also to ultimately get basically all of these values. Okay. So that is just how you solve this 3 variable kind of problem. So in this particular case, if you try to solve it, it would be z equals minus 5 y equals 1 and x equals 7. And then in our earlier cases, we see that the question was asking the regression coefficient for hydrogen bond donor. So in this case, since x was the hydrogen bond donor, we can say, okay, so the coefficient for bond donor will be 7. Okay. So that's how basically you do it. If you get this kind of questions in the assignments and exams, so you have to do it in this way. Now we saw that the answers were x equals 7, y equals 1, and z equals -5, right? So x equals 7, y equals 1, and z equals -5. In the next particular question, we are being asked that if we have a compound with 2 hydrogen bond donors, 4 acceptors, and uh, polar surface area of 3, then what will be the IC50 value? So you have to calculate in the same way. Just remember that x equals 7, y equals 1, and z equals -5 from the earlier question. So you can use these values for donor, accepted, and polar surface area, and then you should get a resultant value. So here we see answer 3. Yeah, basically it will be 3. So if you just have 2 hydrogen bond donors, so 2 times 7 plus 4 times 1 plus 3 times -5. So ultimately you will get the value of 3 nanometer. Okay. So this is how you can solve this kind of numeric problems. Okay. So we just discussed it, but uh, what is the particular element in docking or computing of binding affinity in protein ligand complex that is used for this particular process? So which element is used here for the estimation? So we learned about the 2 important elements in docking. One of them should be the answer to this question. Yeah, I see answer C in the chat. So yeah, scoring function. Scoring function is for calculating or computing the binding affinity or estimating the binding affinity in protein ligand complexes, right? So the search space is kind of the overall conformation space that you have to consider. So the ligand geometry, etc., that will come here also. Search algorithm is the algorithm that uh, basically is used for the search. And virtual screening, of course, it is the overall screening protocol for a large set of library against a particular protein, right? To categorize the binding affinity, however, you need the scoring function to compute the binding affinity. So that was kind of the set of questions that I had. So I have some other slides. I think I might have gone through these kind of slides in some earlier classes. But the overall process of computer-aided drug design is kind of shown here basically. So from the left-hand side figure, so it is actually a figure for a literature from my lab. So one of my seniors wrote this paper. So what she did is that she had the target protein or the target receptor, which is this one, the PDB-ID 60L2 for WNK1. So this is with no lysine kinase 1. And it is kind of widely basically you can say its role is in sort of the heart problems and the blood pressure related problems and so on. So these kind of in symptoms, this kinase has a role to play. And what she did is that she wanted to see whether we can block the catalytic site, which is the active site, or the ATP binding site for this kinase by using certain basically small molecular compounds, right? And here, of course, we can see that the databases she chose, it had 1962 compounds from the library, which is called kinase inhibitory library. Then she also chose the phytochemical library, which had 6412 compounds, and some other MCDB compound library that she also chose. And in total, 10,734 compounds. So that went through a kind of high throughput virtual screening, followed by standard precision, followed by extra precision docking protocol. So this is the protocol that I was talking about, that is employed in the Schrodinger glide. Okay, so in Schrodinger glide, uh, the first 10,734 compounds had to be docked using the high throughput virtual screening protocol, and after that, only let's say the next stage, from high throughput to standard precision, we had like much lower number of compounds, so which could be uh, you know used in a more kind of extensive algorithm, in a sense. So that's why the less number of compounds are kind of docked in a high accurate algorithm manner. And after that, ultimately, she got 6 lead compounds following the ADMET analysis. So ADMET is the prediction of absorption, distribution, metabolism, excretion, toxicity. And then following that, of course, since docking is much less accurate, so she went for MD simulation, followed by binding free energy using this approach MMPPS. And ultimately, she got some compounds, which were basically would be proposed as target molecules. Okay, so that was one kind of example of study that uh, is basically a virtual screening and drug discovery kind of study. So just one moment. The right hand side figure, so this with noradrenaline kinase, it was kind of involved in hypertension related symptoms. So that was the reason that this was taken as a target. Okay. Now here, for example, in the right hand side, we can see that some basics of MD simulations are given. So we have the structures, and here we have the force fields, what we talked about, right? So this force field will have bonded terms, angle terms, and then different non-bonded parameters. For example, electrostatic interaction, van der Waals interactions, and so on. For all of this, we have to calculate the potential energies. So here we have bonded angles, dihedrals, and the non-bonded interaction potentials. And then for by using these kind of potentials, we can basically describe the different atoms and the relation of these different atoms in respect to the others. And what we can do is we can use the Newton's second law to basically see after a certain amount of time where the new coordinate of this particular atom will be. But in that case, we also need to take into, of course, we need to take into consideration all of these interactions that are mentioned here, the bonded and non-bonded ones, because it is not just a free atom is moving in a space freely, but it has a lot of restrictions which comes from the bonded interactions and the surrounding electrostatic van der Waals interactions and so on, right? So if you do that, then you will get a trajectory which will basically give you insight regarding the dynamic behavior of this protein, of this particular protein or this protein ligand complex or any other biomacromolecule in a in your particular set of environment, the environment that you set up for this simulation to occur, right? So it will get you a dynamic behavior throughout this kind of particular state or this kind of environment. So that is how the MD simulation is kind of done. And nowadays, as I talked about in some earlier classes, maybe. So since there is a continuous increase in the size of chemical libraries, so if you want to dock, let's say in a virtual screening processes, if you want to dock a large library such as zinc library or KMBL, something like that, or enamine, something like that, right? So it has billions of compounds. These libraries have billions of compounds. If you try to dock them with a standard docking protocol, so it will take months to years, right? Because you will need a lot of computational power and it will take a lot of time. So that is why currently, since the uh, artificial intelligence, machine learning, and of course the advanced deep learning, since these different algorithms and protocols are kind of flourishing nowadays. So what researchers have basically done in this particular paper, so they have described a procedure which can reduce the time to dock or to screen a large library by a lot. So in this particular one, they have said that they achieved beyond a billion molecules docking with a hundredfold acceleration, and they achieved the results. They get the results within 1 to 2 weeks. So how did they do it? So they basically employed deep learning protocol, which is they're talking about. They have termed this a deep docking kind of, right. So what they do is, of course, from the 2D or from the structure of the ligands, they generate the 2D descriptors. Descriptors. Okay. So these descriptors are basically parameters which will kind of explain some characteristic of the molecule. So here they have certain binary descriptors which all in all together kind of describe the ligand. And what they are doing is that from this ultra large library, only 1% of molecules they are using for training their model. The rest of the molecules, what they will do is that they will not individually dock all of these molecules, but they will predict the score of docking of these molecules from their trained model. So the trained model is kind of like this. So for the molecules they are using in training, they do dock those molecules against the target receptor. They get the docking scores. They convert the uh, ligands into the descriptors, and they train the model by basically correlating that. So they are saying basically if this molecule has this, and these descriptors, then it will have this particular docking score against that protein. Okay. So this type of way they train their model to identify potential lead candidates to basically evaluate the docking score after they train their model. And then what they will do is that ultimately it will be predicting the rest of the library through the trained model basically. So that is one way that the computational time or the basically computational cost and resources can be reduced by a lot. Yeah, I see one hand in the chat again. Please go ahead.
So just to ask whether it's a paid version or free version, AI enabled deep docking protocol.
Yeah, so this is a paper, and I think they have given the associated codes, etc., in their particular paper. So they have mentioned certain softwares which are paid, and they have also given alternatives, if I remember correctly, in this particular paper. So you can take a look at it. So they do have, I believe they have given certain alternatives which are free kind of softwares that you can use for this protocol.
Okay, sir.
And they have also
Thank you,
given,
sir.
yeah, they have also provided certain codes for the workflow that they are trying to propose. So it's
Okay,
quite
sir.
interesting one. Yeah.
Thank you, sir.
Yeah. So I think in the shared Google Drive folder, I have probably uploaded this particular paper. So in the Google Drive folder that was shared, where I put the slides after the classes. So there I have put some subfolders basically. And inside this, one of the subfolders I have put these papers. Kind of, we can take a look at it. Okay. Now, not only do we have the importance of artificial intelligence and machine learning in the docking processes, but through the entire drug development pipeline, we can see, or we can basically use this sort of AI/ML advanced processes to do different tasks. Basically, for example, in target identification, we have multi-omics data analysis, biological network, knowledge graph construction, validation, etc. In drug discovery, of course, we can screen the large libraries. First of all, we can predict the advent properties. We can generate new ligand structures which could be suitable for a drug candidate, and so on. Then in preclinical and clinical trials, we can have diagnostic predictions, then pharmacometrics properties prediction, drug response monitoring, etc., etc. And then after that also, when it is in the final stage, there we also have personalized patient care, personalized efficacy assessment, drug benefit risk assessment, and so on. So in all of these stages, basically we can implement AI ML in a way, which is what kind of currently this the world is kind of proceeding towards, because a lot of advancement in AI ML and its usage in biology, just overall broad area of biology and medicine, that we are kind of seeing from different research groups that are implementing this sort of process, right? So just simple example, for example, here we have database. We can take zinc, we can take GDB. We can take the target database structure database. For that, we can take protein data bank. If you are trying to use the sequence, we can take UniProt, and again electron microscopy database and so on. And then database of drug target interaction, we can take drug bank, binding DB, etc. The binding DB we just looked into in some like a few moments earlier, right? So binding DB database, we can also get the experimental affinity values and so on. Then we can represent our molecules in some sort of uh, way which will be able to be used, let's say, in certain descriptor generation. We can make molecular graphs. Then we can take residue contact map, target structure, etc., sequence. All of this we can take as inputs, and then ultimately we can generate the models, and then finally we can evaluate the models whether it works or not. So ultimately, we can get new drug compounds. Of course, we will need the proper protocols for in vitro, in vivo testing, and clinical trials, and so on. But ultimately, uh, this could again narrow down uh, the identification of new drug molecule by decades, probably. So that's quite good. And then in the synthesis planning and clinical trial processes, also these sort of uh, AI-driven sort of approach is taken or can be taken. So that's what these reviewers kind of mentioning. So I just wanted to put all of this because since a lot of improvement in artificial intelligence and machine learning and so on is kind of coming forward in a sense, so we can properly investigate these. We can try to learn these and apply these techniques in our future research studies. Yeah, I see one hand in the chat. Yeah, please.
Sir, clinical trial. We can proceed with clinical.
Uh, yeah. One second, please.
Sir, like I have some docked molecule. I have done an admit analysis. Every part of it is complete. Can I proceed with the clinical trial myself
No, 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 no,
these days?
no, no, no. You need so that is just the computational part. After that, you need to validate in wet lab, then in vivo studies, and then after that, if it gets passed through all of that, you will need authorization, I think, for from some authorities to get approved for clinical trial. You cannot directly go for clinical trial because, of course, it
Okay.
has patient risks and everything. So you will properly need to
In
get the
between, wet
authorization.
lab has to be done, sir.
Uh,
In between, wet lab has to be done.
yes, yes. So the computational part is only the first part. After that, the drugs, their efficacies, their side effects, they need to be verified in wet lab. And then after that, you have to proceed for example, mice studies. So whether it is showing the results that you want inside mice, whether it is getting any doing any side effects or not inside mice. Then if you get passed through all of these, if your molecule passes through all of these processes, then you can try to ask for some authorization from the higher organizations and so on, and they will approve for the trials maybe if it is passing through all of this. Yeah,
Thank you, sir.
yeah. Okay, so these are kind of the set of slides that I had for this class. So yeah, we will just wrap up the today's class for now. So again, thank you all for joining the class. So please go through Professor Gomia's lectures, the lecture videos, lecture content, and so on, and then please try to attend the other TS classes if possible. And yeah, so thank you so much, and then we'll meet again in the next class. Okay, thank you.